This is a look at a general cell, and right now we're going to be talking about cellular respiration, which is a process that requires oxygen and a food molecule, such as glucose, and gives off carbon dioxide, water, and energy. That's what it looks like in symbols and words. And cells can use almost any food molecule for energy. Right now we're going to take a look at this general cell, and we're going to take a look right at a mitochondria. The first step of cellular respiration is glycolysis. Glycolysis occurs in the cytosol of a cell, which is the water component of the cytoplasm of the cell, where all the organelles and particles are suspended. It's an anaerobic type of respiration, which means it doesn't need air to work. You start off with a single 6-carbon molecule of glucose, and what's going to happen in glycolysis is it's going to split into 2 pyruvic acids. And what's going to happen here is when it splits, it's going to let off energy. And when it lets off energy, things like NAD plus and ADP are going to become ATP and NADH, which all happens from the energy released. Now, you're going to start off the process with 2 ATPs to start off like the whole process. And then at the end, you're going to have 4 ATPs. So you're gaining about 2 ATPs worth of energy. Now, the energy that's left over with the NADH and the ATP is going to go down to the electron transport chain. The ATP is going to stay there. Now, with your 2 pyruvic acids, we're going to go down to the Krebs cycle. And this is a Krebs cycle, which happens in a mitochondria. Yes, in a mitochondria. When introduced to oxygen, the pyruvic acid produced in glycolysis passes to the second stage. In here, pyruvic acids are broken down into carbon dioxide through a series of oxygen energy extracting reactions. The Krebs cycle takes place in the mitochondria, specifically the inner compound called the matrix. Now, the matrix is right inside the inner membrane and the outer membrane, and it's the farthest part in, so all this right here. And then first is citric acid production, and when in the matrix, the pyruvic acid will lose a carbon molecule. That'll just be released into the air eventually once it mixes with oxygen, like CO2. See, that's what plants give off. Now the other 2 molecules will become acetic acid by rearranging, which then joins see, there's the 2 right there, and it joins a compound called a coenzyme A. The molecule is called acetyl-CoA. As the Krebs cycle begins, acetyl-CoA adds the 2-molecule acetyl group to a 4-carbon molecule already present in the cycle, which would be this little guy. This makes a 6-carbon molecule called citric acid. Oh, that sounded really complicated. So pretty much all that happens is the pyruvic acid is going to lose a CO2 molecule, and in this process more NADH is going to be created, and then that 2-carbon molecule, not carbon, sorry. Wait, actually, yeah, it is a carbon molecule. I'm sorry. Anyway, that little molecule is going to get hooked to a compound called a coenzyme called coenzyme A, and when it connects, it's going to be called acetyl-CoA. CoA. I'm abbreviating. And then it's going to get hooked onto this little guy, this 4-carbon molecule, and become citric acid, which is a 6-carbon molecule. And now it is going to go round and around, and each turn of the cycle, which is where the ending 4-carbon molecule takes on the 2-carbon acetyl molecule made from the citric acid production. In the energy extraction, the 6-carbon molecule of citric acid loses 2 molecules of carbon, which is released into the air as CO2. You can see 1 right there, and then 1 right there. Each time it loses a carbon, electrons are added to a molecule of NAD+, which makes NADH, as we've seen this entire time. The electron carrier to make more electron transport chains. So these little NADHs are going to go straight down to the electron transport chain. So it's going to keep going around the circle. It's going to lose that next carbon. It's going to create more NADH. It's going to create some. And with every turn of the cycle, it'll produce ATP. And for every glucose molecule, there's 2 turns of the cycle. So for every glucose molecule, there are 2 ATP thingies made. There's also FAD found in this Krebs cycle, and it's a lot like NAD plus, except it's called FAD. So, and it pretty much does the same thing as NAD+. It just transports electrons when energy gets added to it, so it becomes FADH2. Well, I mean, not FADH2, it's FAD2. And then it'll carry electrons down to the electron transport chain along with all the NADHs. And then finally you're left with the 4-carbon molecule that's going to get hooked to the 2-carbon molecule, acetyl CoA, and the cycle just starts all over again. But now we're going to look at all the NADHs and FAD2s that are going to go down into the electron transport chain, which is the final process of cellular respiration. Now, the high energy electrons obtained in glycolysis in the Krebs cycle produce, in the Krebs cycle process of cellular respiration, are carried by molecules of FAD2 and NADH. The last step of cellular respiration, called the electron transport chain, which is all this little guy, uses those high energy electrons to convert ADP into ATP, or just energy. In the inner mitochondrial membrane, the electron transport chain is composed of a series of electron carriers that pass electrons from one carrier to the next. Kinda looks a lot like the electron transport chain in photosynthesis, because that's pretty much the exact same thing. So, at the end of the chain is an enzyme that combines electrons with hydrogen ions and oxygen to form water. Oxygen is the final electron acceptor of the chain and is essential for getting rid of low energy electrons and hydrogen ions, which is the waste of cellular respiration, which we learned at the beginning. During electron transport, H+ ions, which are all these little thingies, the little dots I made, build up in the inner membrane space, making it positively charged because it has so many positive ions. While the H+ ions go into the inner membrane, they're taken from the matrix, so it's now negative charge because it has a lot less than the inner membrane. Matrix, negative charge. Inner membrane, positive charge. Alright, so as you can see here, all that NADH that comes from the Krebs cycle is coming, and it's going to lose that hydrogen ion, and it's going to become NAD again. And the hydrogen ion is going to go through all these little carrier bobbers. Same with the FAD, H2, it's going to lose that hydrogen ion, become FAD again. It's going to be recycled, like all things do in a plant. And it's just going to go through all these little electron transport carriers. Those electrons are going to run through until it joins with that water, well, the oxygen and the electrons. And then the cell uses a process known as chemiosmosis to produce ATP. The charge difference between the inner mitochondrial membrane means that facilitated diffusion happens through ATP synthase. The hydrogen ions force their way through the ATP synthase, which literally forces the ATP synthase, to spin. With each spin, the synthase grabs an ADP molecule and attaches a phosphate, creating ATP. Alright, so you can kind of see that there. All the little electrons go through here, up to here, get connected with their ions and create more hydrogen and get hooked up with oxygen, which creates water, which is the main waste. And then ATP is created over here at the ATP synthase. So that's what pretty much happens in cellular respiration. You get water and ATP, which is your energy, all throughout at the end of the electron transport, the final process. And carbon dioxide gets put out through the Krebs cycle. So there's all your waste. So you kind of see how this becomes what it becomes, with the oxygen that comes in through the leaf a lot, and glucose that you put in at the beginning, your food molecule. That's what you get out of it. Alright, well, that is cellular respiration. We're good.